Okay, we're recording. Yes. So, thanks for coming. So, this is the second time recording this because I didn't like the way that the first one turned out. Um, but to start off, I want to talk about how we referenced earlier how viruses are able to trick your immune system into attacking itself and attacking its own peptide hormones. So, the example that is well studied and well known of is accomplished through both influenza and coronaviruses, and they both can attack or create the autoimmune response against corticotropin or ACTH, which is a hormone that is released to help release cortisol into the system, and by dysregulating that, you are able to influence the immune response. So, especially for coronaviruses and influenza, if you start making antibodies against corticotropin, you end up having a lot more inflammation, and this ends up making you a lot more symptomatic, which could make you a lot more infectious by making you leakier and making you spew out more juices than you normally would. So one of my concerns is that corticotropin is not the only peptide that is well known to influence immunity. And one of the other ones that I'm concerned about is beta endorphin. So endorphins are working upon the opioid receptors in your body, and your body releases endorphin, and it ends up being an anti-inflammatory and an immunosuppressant peptide. And coincidentally, it is also on the same pro-protein that corticotropin or ACTH comes from. So your body creates a POMC, which is a giant protein, or relatively large, and then proteases come and they separate the bits, or they separate the peptides from that protein. One of those is endorphin, and another one is corticotropin. And so my hypothesis is that since we know that the coronaviruses and influenza are able to generate or make your body generate antibodies against corticotropin, it makes quite a lot of sense that it would also be finding ways to mimic beta endorphins as well. And I have some reason to believe this based on the amino acid sequence alignment between the spike proteins of the coronaviruses and the POMC molecule, where some of the cleavage sites and some of the amino acids of beta endorphin actually line up pretty well with the cleavage sites and the amino acid sequences of the spike protein. So here, because of that, I just want to look into the literature just to see if we have evidence of autoantibodies being produced against beta endorphin, because if that is the case, if it is the case that we are able to produce antibodies against beta endorphin, then there should be somewhere in the literature that describes this event happening. And lo and behold, I was able to find one, and I'm not going to go too in-depth into the paper because I tried doing that in the previous video, and it was a bore. So what I am going to do, though, is just do a quick run-through of what's happening here. So anti-beta endorphin immunoglobulin G in humans. So human IgG antibody specific for beta endorphin was identified by ELISA, or enzyme-linked immunoadsorbent assay, and isolated by affinity chromatography from a sample of 27 subjects. 3 individuals with major depression demonstrated plasma IgG highly reactive with human beta endorphin, while 4 other subjects, 2 with depression and 2 randomly selected blood donors, had intermediate activity. The recognition site for beta endorphin was retained by these fragment antibodies, or antibody fragments. So I'll just do a quick run through on ELISA. So ELISA is a way to measure whether or not you have antibodies or something is producing antibodies against some other protein or some other antigen. And what they did here was they did an ELISA where they coat the plates with protein, the protein peptides, and here they used endorphin, beta endorphin, and for controls they also used corticotropin and another peptide called substance P. And after you coat the plates with the peptide, you then add your samples from the humans onto the plate. And then if the human is making antibodies against that peptide, then those antibodies are going to stick to the peptide, and by sticking to the peptide, they're sticking to the plate. And then you will wash everything off of that, and so all the other antibodies that do not react with the peptides are removed. And then you end up having your human peptide or your human antibodies sticking to the different reactions on the plate. And then following that, you end up adding a different antibody to from, in this case I believe it was from rabbits or goats, and you add it, and the goat antibodies will react with the human antibodies, and then the goat antibodies will have a little enzyme on them that create a chemical reaction if given the proper substrate. And so what you end up doing is you are able to create a plate with a bunch of antigens, and then you can test to see if the human antibodies are present in the samples, because if they are present, then they're going to stick, and then the goat antibody is going to stick on top of the human antibody, and then that goat antibody is then going to catalyze a reaction that you can measure with the machine. And so here they demonstrated that out of the 27 subjects, that 3 of them, or sorry, what was it, 3? Yeah, 3 of them with major depression demonstrated plasma IgG that was highly reactive to human beta endorphin. So these really depressed patients, and not all the subjects were really depressed, but 3 out of them had strong beta endorphin autoantibodies. And then 4 others had moderate. Now, granted, 2 of the other samples were blood donors, and so we have no idea what their clinical reports are, or their clinical diagnoses were. So they could actually have had mild depression. But regardless, there were 2 with moderate levels that also had moderate depression, or that had just depression. So that would indicate to us that out of the people who were depressed, a larger percentage of them showed higher levels of anti-beta endorphin and autoantibody activity. So here we're going to the results, and so here we have the diagnoses of the people that were tested. And so B means that it was a blood donor, so we have no idea what their diagnosis was. And then N is there is a normal control. So this person is normal, and all these people are also normal. And then continuing, where is it? D is patient with major depressive disorder, and P is a patient with personality disorder. So D, all of these Ds are depressed, majorly depressed, and then Ps are the people with personality disorder. Okay, so let's look at this for a little bit. So here we have the anti-beta endorphin activity as measured by that ELISA assay. And here they said that, what was it, 10 through 12 had pretty high, pretty significant anti-beta endorphin activity. And keep in mind, there were some depressed people that had low beta endorphin activity, but there was a significant difference. So if you had depression, you were much more likely to have anti-beta endorphin activity going on. So here we have high activity, all associated with the major depressive disorder. And then here we have moderate activity, so 0.36 or 0.365 for a blood donor, so we have no idea. And then there was another blood donor that they said had moderate activity, so this 0.279 also as a blood donor, so they have no idea what their clinical diagnosis was. And then here we have, for the blood donors, also very low levels of anti-beta endorphin activity. So continuing on, and all the people with no diagnosis, they had low levels. So here 0.112, 0.04, and then 0.057, 0.037, and then all these are 0.0 something. So all of the people who had no medical diagnosis were relatively very low compared to the depressed people. Now, there were some people who had moderate levels. So this person, 0.068, had moderate levels and was depressed. This person had a moderate level and had a personality disorder. This person had a moderate level and was also depressed. So then we went over these severe cases as well. And then this person had a moderate level and was also depressed. So really, if you were to take the depressed group and you were to plot their levels of anti-beta endorphin activity, it would be much, much greater than the normal controls. So there isn't a single, oh yeah, and this person also had normal or moderate anti-beta endorphin activity and was also depressed. So here, I mean, it's a small sample, but I just brought this up to show you that for a very long time we have been aware of autoimmune activity against peptide hormones as a cause of clinical features such as depression or personality disorders. And they go on, and I think I'll go over the discussion because I think the discussion is important to sort of get into the mind of the people that wrote this paper. So we have detected human immunoglobulin with specificity for beta endorphin from 3 patients with affective disorders, subjects 10 to 12, through the use of an ELISA. Subject 10 also had a 1-year history of chronic pain and prior urticarial reaction to hydromorphine hydrochloride. So this person was depressed, but also had chronic pain, and so chronic pain might be an indicator, or is definitely an indicator, of some neurological problem that is going on that is preventing the neurons from firing the correct signals. And this particular person was very unfortunate because as they tried to treat her chronic pain by giving her a opiate, and remember opiates will bind to the opioid receptor very similar to endorphin does. When they gave her opiates, unfortunately, she broke out in she had urticarial reaction, or in other words, she broke out in hives when given this opioid medication. So unfortunately, they had to find some other way to treat her. To our knowledge, this is the first demonstration of an antibody in human plasma with specificity for a neuropeptide that participates in synaptic modulation. The existence of antibodies to central nervous system neuropeptides and the recent isolation of antibody to serotonin receptor from autistic children, and this is contested. I don't remember which paper it was. Keep in mind this paper is pretty old, so we've definitely come a long way from this paper. But this is the serotonin receptor, and if you have an autoimmunity against that serotonin receptor or parts of it, that was implicated in autistic children, or at least a subset of autistic children. And I believe that it's still true, but it's a lot more nuanced than just that. And this is suggesting the possibility that synaptic central nervous system function may be influenced by immune mechanisms. The extent to which this occurs in normal individuals or is pertinent to psychiatric disorders cannot be answered by the data presented here. Conclusions about the potential clinical role of these antibodies in the mechanism of affective disorder will require studies of larger patient and control populations. So disturbances in the appetite and in the endocrine and circadian rhythms comprise principal features of affective disorders. The circumventricular organs which are some of the parts of the nervous system in your brain that are basically do not have the blood-brain barrier. So it's easy for them to produce large peptides, and those peptides can get through into your bloodstream or get into your system a lot more easily than other parts of the brain. So the circumventricular organs, in particular the median eminence and the pineal gland, are central nervous system structures implicated in the regulation of endocrine and circadian rhythms. These central nervous system sites are highly vascular and contain high concentrations of neuropeptides but lack a blood-brain barrier, thus permitting the passage of high molecular weight proteins such as immunoglobulins. So here they're saying that because these organs and tissues are not as protected as the rest of the brain, which has the blood-brain barrier, um, they participate in creating peptides and secreting them, but likewise they are also more prone to attack from antibodies. So normally antibodies wouldn't be able to pass through the blood-brain barrier, but since these organs do not have the blood-brain barrier, then uh, they can be attacked, unfortunately. This combination of factors makes the circumventricular organs candidate sites for immunomodulation of neuroendocrine and synaptic events. So here they're focusing their attention just on the nervous system, but you make a lot of beta endorphins in other parts of the body. So you really should not just be we should not just be focusing on what are the neural implications of this. We should be focusing on, well, any tissue that both produces beta endorphins and has receptors or has opiate receptors and requires the beta endorphins to function correctly, those are all tissues and cells that are implicated in being susceptible to autoantibody attack, or at least being dysregulated from autoantibodies. Continuing, antibodies to neuropeptides conceivably could penetrate these sites and alter endocrine and circadian function. Such antibodies could potentially spread by anterograde and retrograde exoplasmic transport, which is basically moving large proteins on is it the microtubules or the X? They're basically moving them through the axons of the nerves. Exoplasmic transport, that's in the name. And if antibodies to beta endorphin and other neuropeptides are present in the normal population at meaningful levels, they might potentially yield interactions between immunoregulatory and neuroregulatory mechanisms. So here they're saying they were able to detect low levels of anti-beta endorphin throughout the normal population as well. So none of these numbers were zero, right? They're low. Oh, wait, that's not the right one. So none of these numbers were zero. They were all, but some of them were low, but they were not zero. So there is some activity that is going on. There's always some anti-beta endorphin activity that your immune system is doing. But you do not want that to become dysregulated or get too high, because you might be pushed, or you are going to be more likely going to be pushed into the category of depressive if you have higher activity of anti-beta endorphins. Here, so the occurrence of elevated levels of antibodies to neuropeptides. Oh shoot! Before I go, they're also saying that maybe this is an important part. This is of a healthy individual's regulation of their neuropeptides. So maybe having healthy levels of these autoantibodies might actually be playing some sort of regulatory role that keeps you healthy. Possibly. That's just a presumption, because it seems to be conserved among people. So you would assume if it's conserved, it might not just be noise. It might actually be serving a physiological role. Whether that's speculation, though. Maybe it's all negative. Maybe it truly is completely negative that anyone makes any antibodies against beta endorphin. That is also a possible scenario. And maybe by controlling or completely eliminating this autoantibody response, maybe there is a net positive to the human population by doing so, but we don't know this yet. So, or at least this is a really old paper, so maybe we know this by now, but I haven't seen any papers indicating that. Anyways, the occurrence of elevated levels of antibodies to neuropeptides in patients with affective and related disorders provide preliminary data that suggests that disordered immune network regulations may result in autoantibodies capable of inducing selective modulations of certain neuron groups and their projections. Such antibodies could potentially provide compensatory immunoregulation responses to a disturbed immune system, central nervous system interaction resulting from a primary neuroregulatory disorder. So stepping back, and nice, only 20 minutes. So stepping back, we know that people are generating these autoantibodies even in healthy individuals at low levels. What is something that can knock that system out of place or out of whack? And one of those things would be the generation of, if you have an immune response against a virus that is producing an antigen that looks similar to beta endorphin, then it is possible that your body starts upregulating the production of the immune cells that are reactive against that virus, but then simultaneously are active against beta endorphin. And so we do know that this is a strategy that viruses do employ to confuse your immune system, because if the immune system is making antibodies against the virus epitope, but they're getting sequestered by beta endorphin in the body, then the virus is able to live longer, or the virus is able to duplicate longer because those antibodies are getting sequestered by other peptides in the body. So what I would like to present is that it is possible that viruses are playing a very significant role in affecting peptide levels, not just during their infection, but long after, because your autoimmune response is pretty difficult to get back down to normal levels. It takes a long time for your body to ramp down from an autoimmune response, because what's making it ramp up in the first place is the presence of that peptide. And if that peptide's as ubiquitous as endorphin, then it'll be very difficult for your immune system to stop recognizing it and to stop duplicating the immune cells that are designed to attack that particular epitope. So, what else do I want to say? Just, another thing that I want to mention is that there is a possible there's a possible role in autoimmunity for all of this that is not just against beta endorphins. Beta endorphins are beneficial in that they help suppress the immune response. And I was reading some other papers where if you have a dysregulation or lower levels of beta endorphin, then that could end up making autoimmune diseases worse. It's as if the body wants to remain inflamed more. I'll probably make my next video about this, but there's an example of a woman who had an autoimmune disease. I believe it was Crohn's or some sort of gluten intolerance. And so she had both intestinal problems and also on top of that skin, like it was some sort of rashes or hives that she had. And in order to get her immune system under control or get the inflammation down, they removed gluten from her diet, which really significantly helped with the intestinal problems. But she still had some of the skin condition that had persisted for years. even after the gluten-free diet. So what they ended up having to give her was an opioid blocker, but it was a very short-lived opiate blocker. And by doing so, her body started producing more beta endorphin in the gut. And that beta endorphin that was produced and helped suppress the immune system more, which allowed her body to heal from the rashes from her autoimmune disease. So what we could imagine, what we could easily see is a runaway effect, right? So if the body starts producing autoantibodies against beta endorphin, then the lack of endorphin that is running through your system will then make your body a lot more inflamed, or a lot more active, at least immunologically wise. And then when that happens, it starts producing more antibodies, which could sequester even more beta endorphin. And you could see how this process could easily create a negative runaway effect down the road. So this could have implications on the multi-system inflammatory syndrome that we are experiencing with COVID-19. And that somehow your peptide hormones or your regulatory elements are getting out of whack, and there's probably some positive feedback loop that is pushing people closer and closer to being in a hyperinflamed state. So diseases, or I should say the peptides that are important to look at would be, the corticotropin, which manages cortisol levels, and cortisol is the anti-inflammatory. So if those corticotropin levels end up getting sequestered by antibodies, then your cortisol levels go down, and then you become more inflamed. So that's one positive feedback loop that we could be seeing. Another one is the beta endorphins. So beta endorphins go down, you get more inflamed, and then you get more beta endorphins being sequestered by antibodies. So that would be a different positive feedback loop that would be of concern. And then one of the other ones that I talked about was prolactin. So not prolactin explicitly, but there are a few peptides that are able to suppress prolactin. And if you are suppressing those peptides, then you have an increase of prolactin in the body. And if you have an increase of prolactin, that could have some pro-inflammatory effects as well, which could then result in the release of more antibodies, which would then result in the release of more prolactin. And you can see that's another possible positive feedback loop that would be difficult to manage if the system goes completely out of whack. So what else did I want to say about that? So I just want to say those are three possible mechanisms of immunomodulation that could go out of whack just from the possibility of viruses imitating or mimicking your own peptide hormones. And so I did do a sequence alignment of the entire POMC protein, and some of those sequences aligned pretty well with beta endorphin. So now my concern is beta endorphin, like corticotropin, might also be attacked, or might also be one of the molecular mimics of the spike coronavirus protein. And not just of SARS-CoV-2, but the other SARS and the other coronaviruses as well. Granted, though, those other coronaviruses are not eliciting as strong of an immune response. So they're not going to be in as high the viral load that you're going to get from the other human coronaviruses that spread around every single year. You're not developing as strong of an immune response against those coronaviruses. So theoretically, hopefully, the results or the long-term immune consequences of getting those coronaviruses will be significantly less. So if your body is able to downregulate its own immune system right away after contracting a normal coronavirus, then that would indicate that it's not creating these long-term immunomodulatory effects that I personally am worried about. So, yeah, in regards to MERS, SARS, and SARS-CoV-2, since those are able to create a much, much, much more significant immune response, we're probably going to see a lot more serious um, or a lot more persistent immunoglobulins or antibodies being formed against them. And how those antibodies affect your metabolism in the long term is something that, frankly, I don't, how do I phrase this? I just don't want it. I don't want to have to deal with that type of serious wrench being thrown into my metabolism. So, um, yeah, that's all I really wanted to share with this video. I think my next video, I'm going to go over the other paper with the women who had the who required the endorphin antagonist as a treatment protocol for her autoimmune disease. But yeah, so if there are any questions, the link to this paper is down in the description if you want to look at it yourself. Obviously, it's old, it's probably outdated. I just wanted to have some example put forth for my 4 subscribers so that you could see that this is not something that just came up within the last few years. This is something that has been known about for decades. So with that, thanks for watching. I will see you within a few hours, I think. So thanks.